introduce myself. I'm Denis Wirtz. I'm a faculty at Johns Hopkins University with appointments in chemical engineering as well as oncology and pathology. And as I said, as a team of faculty, student, postdocs, research faculty, we've together brought and developed a suite of tools that are now amount to a very large set of tools and very versatile in its applications, addressing the challenge of mapping out human tissues, which is always extra difficult compared to maybe tissues harvested from mouse models or animal models in general, in that they're harder to get, in that there's additional biological challenges. But CODA really tries to image large volume of tissues, and we're talking about multi-centimeter cube of tissues, and yet preserve submicron resolution. So if you do the math, that's 6 or 7 orders of magnitude. We'd like to be able to look at whole tissues and their overall anatomy, start zooming to the microanatomy of different parts of these tissues, like blood vessels, maybe ducts, maybe sweat glands, maybe islets of Langerhans, and then further zooming in, look at clusters of cells within these substructures, things like immune cells, things like senescent cells, stromal cells, and then further zoom in to try to look at, for instance, the transcriptomic profile or proteomic profile. And we've developed to do this an AI-based tools so as to be able to automate the different tasks that goes into this mapping. We trying, and this has been part of our goals, to minimize human intervention, so that any additional work that needs kind of to do it, by hand cannot be automated thanks to this AI tool. I'm gonna stop here and have Professor Wu introduce himself.
Hi, thank you, Denis. My name is Pei Hsun Wu. I'm also from Johns Hopkins University. I'm a research associate professor at the IMBT Institute for NanoBioTechnologies within the Whiting School of Engineering. So as Denis described, our group is within the SenNet. Our main, we serve as the technology development and application centers within the SenNet, and with our projects, it's be mainly to leverage the CODA tool, the tool that Denis just described, to build the 3D spatial multi-omics analysis to map the cellular senescence directly in human tissue in 3D. We also, in addition to CODA, thanks to challenging with the mapping senescence cell states in human tissues, we also established highly sensitive multiplex imaging platform for detect cellular senescence in FFPE archived tissue samples. And so our role in SenNet is generally trying to generate spatially integrated volumetric map of a senescent cell in human tissue in situ to really define how the senescence vary across different tissue types, compartments, ultimately contribute to our better or comprehensive understanding of a human senescent cell, atlas in human tissues.
In a way, the genesis of CODA was initially trying to address a challenge posed by one of our colleagues in the Department of Pathology, Professor Ralph Ruben, who really thought that maybe an AI tool could detect rare events, and he has his eye on trying to potentially automatically identify precancerous lesions in tissues in general. The problem with traditional pathologies, typically a pathologist will diagnose a disease based on 1 or 2 sections and grade a tumor, maybe assess immune cell infiltrate with this rather limited sampling of the tissue. But if you're trying to identify much rarer cell clusters or cell populations, it is more and more clear that a 3D assessment is the only way to go. And so what we set out to do, and really an effort led by Pei Hsun, was to try to turn this 3D imaging challenge into a series of 2D imaging challenges. And the idea was to take a tissue, maybe a tumor, maybe a whole organ, maybe even a whole organism for that matter, serially section it, and use very standard stains like hematoxylin neosine to train an algorithm to automatically identify different tissue components, such as blood vessels, duct, acini, and different other structures like precancerous lesions. And instead of doing it by hand, one section at a time, and then try to realign these different sections somehow, Pei Hsun took on the technical challenge to do this automatically and indeed use modern tools of AI to do so, but also developed additional tool to automate, for instance, this alignment step in the CODA workflow. That is, if you have a series of tens or hundreds or thousands of sections, could you feed images of those sections into a computer to properly and quite faithfully register, align these different sections? And long story short, now it's done automatically, again using additional AI tools. So think of senescent cells. They're going to come in clusters. They're going to be hard to detect if you're doing just one or two sections in whole organs or in a whole organism. That becomes a lot easier to identify and to position in that tissue using a 3D imaging tool. Before CODA came online, uh, either as a pathologist, you had to go for volume. So think imaging technique like MRI, CT scan, PET scan, right, where you have the contours of an organ, the contours of a tumor, but you don't get this high single cell resolution you're seeking. Or you use a high resolution microscope, then you get the cellular, even subcellular resolution, but then you don't get the whole larger perspective of where these cells belong to maybe a set of cells like a cluster and them cells belonging to an organ. So you don't have the whole spatial context of these cells you're interested in. But here, our team was able to, in a way, have it both ways. Large volume assessments, even though we're pursuing this very high spatial resolution at single cell.
So I think our CODA, how it contributes to senescence. So I like what Denis said, right? The tissue, the human tissue is a 3D structures, and a lot of our understanding in cellular senescence has been primarily at its background focus on more like a cell culture model, and also in the 2D section slides, right? You can see kind of roughly there's senescence signatures in 2D and in culture model, but really not a lot of understanding has been, understand in the human tissue in situ level. I think that's where kind of CODA we see the CODA, how the CODA can bridge this gap and then bring really like vital information about the cellular senescence in situ with it. So therefore, we propose this idea with the SenNets, and I think Review also like this idea as well to really fully use a 3D volumetric way with leverages the CODA to under cellular senescence very well. So our proposed project is actually quite straightforward. We, with the serial sections technology, there are a lot of possibility, right? Because I think one big advantage in the CODA workflow is we don't need all the tissues to do the structural mappings. We just need to usually every 1/3 of tissue or of one-third of the tissue in serial used for the histological staining and for the tissue structural mapping. So leave the other 2/3 of tissue will be ideal material to do other integrate with other omics analysis approaches, such as the multiplex immunofluorescence or spatial transcriptomics. So this way that using the CODA serve as a backbone, we can really integrate various technologies to provide 3D context, and we also propose using the multiplex immunofluorescence and a workflow to really map high content senescence markers in the human tissues as well. And then these together offers to really to map not only the 3D structures, tissue 3D structures, but also allow us to really locate where the senescence potential niche is within this the human complicated human tissue structures, and then from there we can potentially even further zoom in to find out what's applied with integrates other spatial assays to really narrow down to see what the transcriptomic sectional differentiated from this. So I guess that's the I mean it sounds already quite exciting to me about that. So I guess that's kind of how we kind of apply our CODA with the SenNet's missions.
And I think in a way, what patient just described illustrate really well the power of CODA, right? We talked about how it can examine large volumes of tissues and yet preserve this very high resolution, which is typically not done with the same instrument. But there's a third aspect to CODA that is really makes it very versatile. Any advance, technological advance, where one can examine maybe the molecular profile or the proteomic profile or transcriptomic profile or metabolomic profile of a tissue, but only using a 2D assessment, can now be expanded to 3D. And here, Pei Hsun developed a way specifically to identify markers of senescence, and that was already a technical challenge that he has now solved, but now can be expanded to do 3D mapping of senescent cells in 3D, marrying and exploiting and leveraging all the benefits of large volume, high resolution, and even integration of cutting edge multi-omic techniques, such as those recently developed by Pei Hsun.
Think it can have a lot of implications. I mean, impacts. First of all, I think mainly as a tools, right? Our original proposal to study senescence in our proposal is focused on pancreas, ovary, and breast tissues. But the technology itself can be easily translated to all different tissue, and which can be really, I think, merged very well with all data or sample generated from the SenNet consortiums. There are so many of them, right? So they already have some, maybe they have a lot of 2D overview of the senescence signatures they might find. I think this is called a platform over like really great opportunities to really try to really translate those signatures they might find into 3D on the whole tissue, whole body understandings, how actually spatial distribution look like. So I think at least to me, one big contribution the CODA potentially can have to the SenNet community and the consortium is as a tools that's can be really used, and we'll hope to see that to adopted by the other groups and the centers to really use this technology to really enhance our understanding of senescence in across tissue types in humans.
I'd like to add that one thing that Pei Hsun and I have really thought quite deeply about is how to make those tools available to the larger community. And all the source codes are available on GitHub, which is unusual in this field. Too many, I'm afraid, of our good colleagues do not do this, do not share the source code. We do. We also have now published a series of very detailed protocols to really guide those adopters of the CODA workflow to then maybe customize that workflow for their own application. But at least they're not starting from scratch reading formal scientific papers without the necessary details that would allow them to adapt these technologies for themselves. It's not a trivial workflow, and we recognize this. That's often the case of any new technology. But from the get-go, and we are really doubling down on this, we're ensuring that those protocols, those tools are immediately available to the wider community of scientists and engineers interested in senescence. We're gonna look back at this and see that the only way to properly assess the heterogeneity, the complexity of human tissues is to look at them, examine them in kind of molecular depth in 3D. It's going to be, is absolutely the future for sure.
But I think maybe one thing to just to highlight a little bit more is like CODA is not just a tissue mapping tool; it's also integration tools, right? So it's like I think it's just a workflow as a backbone to bring a lot of technology potentially together. It's developed not only commercialized one, but potentially the one developed within the SenNet consortiums. It just through a series of sections schemes a lot of materials like leaving there unused material can be really reuse it and bring it all together in the same special coordinates to really help engage our understanding in the cellular senescence.